Hello students, welcome back to my channel CSIR Buddy, and this is the part 1 for eukaryotic replication. So first starting with the basics of replication. So when we talk about replication, what does it mean? Replication means when we make a copy of DNA from the parent strand of DNA, then this process is known as replication. And why do we need replication? Because when in the time of mitosis, when the cell divides, we need equal distribution of genes, or for that matter every organelle, we need organelle to be distributed equally in both the cells. So during the time of cell division, the DNA replication occurs. Now cell division undergoes a cell cycle. So there are some of the events that are included in cell cycle. So the very first phase of cell cycle is the G1 phase, that is the growth 1 phase. Then comes the S phase. In this particular phase, the DNA will replicate. And then comes the G2 phase. Again, this is also the growth phase. And then comes the M phase, that is mitosis or meiosis phase. Mitosis or meiosis. So you will learn about this cell cycle when you study about cell biology unit. When you will study about cell biology unit, then only you will get to know about the cell cycle. But for now, this is important that cell DNA replication occurs in the S phase, and the cell cycle is regulated from CDK. CDKs are present in this process which interact with cyclins, and this process gets continuous right of the cell cycle. So the point was that the DNA replicates in the S phase of the cell cycle. Then after S phase, the G2 phase comes, and then at the last we get mitosis or meiosis, and then we get number of cells, that is one cell divides into two. Now more information that the DNA replication is of semi-conservative nature. As I said before, that the replication DNA replicate from the parent strand. So semi-conservation means that one strand will be as it is the parent strand. When the DNA undergoes replication, then what will happen? The two DNA which we will be getting. So in those DNA, one will be the parent strand as it is, and the other will be the new strand. So this is what is known as the semi-conservative nature. So this was the parent strand, and instead of getting both the strands that are new strand, we only get one strand, that is a new strand, and the other strand is conserved, that is the parent one. So that's why we use the word conservative. So the parent strand is conserved over here. So we get two copies. Of course we get two copies, but in each copy one of the strand remains the parent strand itself. So I think this thing is clear till now. Now moving further, if we make a DNA molecule, so we all know that DNA molecule is made up of pentose sugar, and then we have a nitrogenous base, then H and OH. The important thing is that 5' is loaded with phosphate groups and 3' consists of OH group. Now what happens, the other nucleotide comes, and now this is the time of bond formation. So now we will learn how the bond is formed between these two DNA molecules, or we can say nucleotides. What happens is that this OH group, the O group, does a nucleophilic attack on the phosphate group and breaks the bond, and we get 2 phosphate group out and 1 phosphate group remains intact, and these both make a bond, and the bond which they make is known as phosphodiester bond. So this is how the phosphodiester bond is made up. Now this N group will make a bond with another nitrogenous group, and as we know that there are 4 types of nitrogenous group present in DNA, that is adenine, thymine, guanine and cytosine. So A makes double bond with T and G makes triple bond with C. So this is how the nitrogenous pairing is done, and as we all know that DNA molecule is anti-parallel. That means one strand will be running 5' to 3' while the other strand will be running 5' to 3'. As these are anti-parallel, the replication which happens, is bidirectional. And why bidirectional? Because the primer synthesizes the DNA molecule only from the 5' end to the 3' end. So in this one case, the primer will be attached over somewhere over below where this 3' is set. So 5' to 3' synthesis will go on. In this case also, the primer will attach somewhere over here from 3' to 5'. So that's why this replication is a bidirectional replication. So I think this much the basics are clear to you all. Now let's get started. So this is the introductory part. DNA replication is a process of producing 2 identical copies from one original DNA molecules, as we have already studied. It is the basis of inheritance and therefore it occurs in all living organisms. So it is a basic of inheritance because the DNA molecules constitute of genes which are inherited to the or passed on to the offspring. Fundamental mechanism of DNA replication is same. However, there are some variation due to difference in molecular properties. DNA replication in eukaryotes is more complex than the DNA replication in prokaryotes. So eukaryotic DNA replication is complicated then as compared to prokaryotes. DNA in eukaryotes are highly condensed nucleosomal structure which needs to be unwound before replication. Highly condensed nucleosomal structure is present because DNA molecule is a very long fibers, and those long fibers couldn't be handled by the nucleus. So they cluster up in a form of nucleosome. So they make a clusters, and these proteins which are allowing the DNA to wrap around it, this protein name is histone protein. histone protein. So when the positively charged histone protein interact with the negatively charged DNA molecule, then they make a condensed structure that is known as nucleosomal structure, or else we can say nucleosome. Majority of DNA replication synthesis occurs during S phase, as I have already discussed, of the cell cycle. Telomere is synthesized in late S phase. Due to complex nature of eukaryotic chromosomes, replication is not as well understood as that in prokaryotes. Now, because of the complicated nature of eukaryotic chromosomes, eukaryotic replication is not yet understood so clearly. Then we have been understanding the prokaryotic replication. Replication is bidirectional as in prokaryotes. So both in pro and eukaryotes the replication is bidirectional. So this was some of the information related to replication. Now going further. If we talk about initiation, G1 CDK is activated by cyclins. So what is CDK and cyclins, you will learn in the cell biology unit. Activated G1 CDK activates S phase specific CDK. So CDK and cyclins are something which regulate the cell cycle. So CDK activates S phase specific CDK. So S phase specific CDK are activated because in S phase only the DNA replication takes place. Activated SCDK then triggers the assembly of proteins at the origins of replication. So the origin of replication, when the SCDK gets activated, it will trigger the assembly of some of the major proteins which will help in replication in the region of origin of replication. Nearly 10,000 and 1 lakh replication origins may be found in a dividing human somatic cells. So these many replication origin could be found in a dividing human cells. Each origin must initiate once only during each replication cycle in order to avoid duplication of DNA segments that have already been replicated. So this is very important. Although it is having many multiple origin, but only one origin will only one replication at time will be taking place, because in order to avoid the duplication of DNA, right? So this was about the initiation. Now moving further, after SCDK activation, SCDK transfers a phosphate to SLED2 and SLED3. So I have shown this in the diagram. SLED1 and SLED3 and SLED2 are getting phosphorylated. Phosphorylated SLED2 and SLED3 bind to DBP11. So this makes a bond with DBP11 as shown in the figure, which acts as a scaffolding protein that holds the replication origin proteins in position. Now this protein will hold the replication protein in their position. After that, CDC45 associates with the origin of replication to form the preloading complex pre-LX. So CDC45 is working as a preloader. Then a large number of different proteins such as helicase initiates unwinding of DNA helix. So now helicase, what does helicase does? That it unwound the DNA helix so that the replication could be moved further. ORC recruits CDC6 and CDT1, also known as actor. CDC6 and CDT1 recruit the mini chromosome maintenance. So MCM is the helicase for eukaryotic cells complex to form the pre-replicative complex. So now MCM, CDT1 and CDC6 recruit MCM so that it can form the pre-replicative complex, also known as pre-RC. So in the case of eukaryotes, we have seen that MCM is the helicase, but in the case of prokaryotes, we saw that DNA B was the helicase and DNA C was its loader. The MCM complex consists of 6 proteins MCM2 to MCM7 that form a hexameric ring around the DNA as shown. This is the MCM helix and it will be making a hexameric ring around the DNA. After activation, MCM acts as a helicase to unwind the double helix and thus is equivalent to the bacterial helicase DNA B, as I have mentioned over here, that it is equivalent to the prokaryotic DNA B. So you have to remember these many things, that MCM is equal to DNA B and the CDC6 is equal to DNA C. So this is the helicase and this was the helicase loader. So you can correlate from this. Now origin recognition complex, this is 6 polypeptide, that is CDC6. Then CDC6 ATP bond, ATP is used, this is CDC6, and then MCM comes into, and MCM is recruited by the CDC6. So this is the formation of pre-replicative complex with 2 MCMs, that is MCM2 to 7 and MCM CDC6. So this makes the pre-replicative complex. So that was it for this video. If there is any doubt regarding this video lecture, so please mention this into the comment section, and if you like my channel, please like, share and subscribe to our channel, and thanks for watching till end.